So I think that one important concept that is emerging in the last years is really this concept that resistance is not just driven by genetic mutation, but there's a lot of other mechanisms that they can also modify the sensitivity of cancer to therapy. In the past, a lot of emphasis in this particular setting has been focused on the tumor microenvironment. And it has been shown in several different types of tumor, starting from leukemia, breast cancer, colon cancer, that the tumor microenvironment, by secretion of a cytokine and modification of the matrix, can actually modify the sensitivity of tumor to therapy. More recently, it has also been shown that cancer are much more diverse in their composition, and there's also diversity that is caused by epigenetic mechanism. In other words, the cells, despite having the same genetic makeup, they can essentially change among different cell states. That is driven to mechanisms that mostly are to epigenetic stochastic alteration. And when the cells, they move from one state to the other, they acquire a different phenotype. They completely rewire their signal transduction, and so they change the dependency. So in other words, if we target, let's say, EGF receptor in tumor that they are driven by specific mutation in EGF receptor, a particular subset of these cells, they will not be addicted to this oncogene because they will have activation of other signaling pathways that they are parallel. And this modification of the signaling network has really been pioneered by Neil Rosen and Sloan Kettering, in which it really has been shown that the treatment, inhibition of a particular pathway can essentially activate parallel pathway, because again, like the signaling network within the cells is highly complex. And during evolution, our cells, they have been evolved to really have a very robust signaling. So in other words, if you block one pathway, you can always find something to compensate for that. It has been shown indeed that cells are, especially cancer cells, are highly plastic and they can switch among these different cell states. There's a different cell state that they have been described. Some of these are really, again, like these epigenetic driven cell state. And what is interesting is that some of them, they acquire essentially characteristics that they are typical of cells that that they are part of tissue that they are undergoing repair. So this is sort of a fascinating concept because it can also argue that maybe tumor are wound that never heal. And so there's a subset of cells that they are essentially in this cell state that is characteristic of epithelial cells that they are undergoing healing. Another very interesting subset of cells that again like represent these plastic cells are what they are usually referred to as persistent cells. And they have been described initially in a cell culture, and some more recent data they show so that maybe they can be relevant also in tumor. The difference among these different subtypes is that while the first one, they occur sort of naturally, in the other case, it's really more like a stress response of the cells. So in other words, if you treat cancer epithelial cells with a drug, some of these cells, they do sort of in a stress response, and they change, and they become more resistant to apoptosis. But this cell state, stress-induced cell state, is really transit. So as soon as you remove the drug, the cells, they return to a normal state. And this is, again, like, is very interesting because conceptually, the way that we can treat them, the tumor based on these different cell population is different. Because in the case of the persistent, you can maybe just oscillate the drug until a point in which you will generate an exhaustion of these persistent cells. In the other case, instead, these cells are just present in the tumor. So in this case, it doesn't matter if you are removing the drug, they are still there. One interesting observation that we have made in my lab recently is that some of these cells that they are epigenetically driven and they are already present up front in tumor naive for drug treatment, they have an intrinsic defect of repairing DNA. And this is particularly interesting because it's arguing that this particular subset of cells, they are highly diverse from a genetic point of view. So in other words, you have a tumor cell that they can change cell state. And when you acquire and pass to this other cell state, now it can accumulate more mutation, in particular copy number alteration and haploidy. Therefore, it generates a variety of different cells, different sister and brother. So this is interesting because many years ago, actually at Constance Harvard lab, it was described by Luria and Delbruck, the concept that in this case, bacterial population, if they are more diverse they are genetically, and more essentially they have a probability to become resistant to bacteriophage. So in this case, it would be the same. So you increase the genetic diversity of a population, and so you increase the chance that there will be one particular clone that will be able to sustain drug treatment. So this is exciting.
Very evolutionary.
This is very evolutionary. So this is why biology is very interesting, because you have a lot of different fields that they are merging, and you can utilize knowledge acquired in a particular field and transfer it. This is why AACR is great for this, because you can just move from one room to the other and you can really be exposed to a completely different field. The reason why I think this finding of these non-genetic mechanisms of resistance are important and they are exciting is because it's changing also the paradigm. Like, for example, in the case of target therapy, in the case of lung cancer, and lung cancer driven by mutant TGF receptor, in this case, like 10 years ago, we identified this mutation. We know that patients that they have this mutation, they are highly sensitive to inhibition with a particular drug that is an EGF receptor inhibitor. We know that after 1 year, the 50% of the tumor they relapse, they have a particular mutation that is blocking the interaction with the drug. And therefore, pharmaceutical company, they have developed second, third, fourth generation. So with the idea is that you can continue and so progressively change. So if we are correct, and we are saying that now is if you have these cells, they are present up front, then you essentially what you are doing, you are just selecting sort of for an increased heterogeneity of cell population. Therefore, with time, you will acquire resistance much faster. So the way to do it is, rather than to wait for the occurrence of this mutation, is to really try to wipe out the tumor cell population as much as possible. So rather than to do a sequential treatment, what we are suggesting is to instead to do a combinatorial treatment starting with. And this is really resembling a paradigm developed, for example, in the case of bacteria with antibiotic, or in the case of HIV, in which they clearly show that a combinatorial treatment is much more effective than a single dose or, sequential. I think it's important to understand the molecular detail. So what is driving this cell plasticity? What are the mechanisms? What are these cells dependent on? So in other words, to really characterize this better, in the case, for example, of cell plasticity. Because in this manner, then we can specifically target and we can design better combinatorial treatment to really combine these. And then, of course, it would be nice to start really to have clinical trial and to really try to see if this idea is correct.